All right, so today I would like to introduce to you GDC 2.0, a new cohort-centric design. My name is Bill, and I am the lead for GDC User Services. And let's get started with this presentation. So most of this presentation is going to be a demo, but there's going to be a bit of an introductory information about the GDC 2.0. And at the end, there's going to be time for questions from the audience. So if you have any questions, feel free to type them into the Q&A panel. There's another chat panel. I wouldn't recommend typing into there, but the Q&A panel will see anything that you ask there. So we'll get to those questions at the end of the presentation. And yeah, why don't we jump right into it? So you've heard in the title of this and the all of the different, all of the different headers in this presentation. The name cohort. So here I like to define what a cohort is, and it's a group of cases, usually referring to patients when it comes to the GDC, that share a set of characteristics. The new GDC 2.0 workflow separates the cohort building process from all of the other functions of the portal. And this is something we're going to demonstrate very much in this presentation. So separating these two processes allows for the same analyses to be performed on the same groups of cases across all available GDC tools. So here this is a GDC 2.0 workflow, and it can be broken down into two basic steps. The first step is building a cohort. This is going to be demonstrated right after this. But this is just the act of taking all of the cases in the GDC and filtering it down to those with the clinical and biospecimen attributes that you're interested in. The next step is to take that cohort and use it with the tools in the GDC Analysis Center. These are all of the available analysis tools, and the tools will automatically be applied to your cohort. So when you go to a tool and you have a cohort, they are automatically applied, and you're going to see that in a bit. So that's really the end of the introductory information. Like I said, this is going to be a mostly demo presentation. So let's go over to the actual portal and take a look. So here we are at portal.gdc.cancer.gov. And a first thing I want to demonstrate is creating a cohort. So one can create a cohort by going to the cohort builder icon, third from the left at the top bar here. So once we click on that, there's a few things I want to bring to your attention. First is that we have an unsaved cohort. That is our current active cohort. This icon suggests that the changes have not been saved yet, so we will need to save it. And the number of cases on the right here, this will update as we filter our cohort down, but right now it has 44,451 cases, which are all of the cases in the GDC. So our goal, and that's the format of this demo. We're going to have small goals for most of the different tools, and these will be used to show the basic functionalities of these tools. Not every single thing that GDC 2.0 will be covered. It is way too much. But we recommend checking out some other documentation sources that we're going to show at the end of this presentation. And there will be subsequent webinars on different features of the GDC. So our first goal is to use the GDC 2.0 cohort builder to build this particular cohort. So the first property is that we want only cases in which the primary tumor is from the kidney. And we can get to this in the general category here under primary site. I'll click on the magnifying glass here on the top bar and type in kidney or partially kidney, and that comes up and I can select that. Um, so now that I've selected that, you can see at the top here, we have much fewer cases. That is because it is only a subset. And as I click on each of these filters, they will be recorded in the query bar here. So the query toolbar records what your query is. And this will carry with you throughout all of the different parts of GDC 2.0. So our next attribute here is that we want only patients that were diagnosed between ages 50 and 60. So we can get to that in the demographic category on the left bar here. Down at age of diagnosis. And there's the opportunity to change between years and days and customize this, but we only want between 50 and 60 years old at diagnosis. So we can go ahead and choose this radio button. And this brings us down to 450 cases. Next, we want only cases in which the gender is male. So we'll go ahead and click on male here. And the last one is that we want only cases that have whole genome sequencing data, WGS data. And we can find this filter down in the available data category at the bottom. And at the top right, experimental strategy is our property of interest. So we'll go ahead and click on 6 more because it's not like the primary site, which had many different values. We only have a few. So we can select WGS from this list. And you're going to see this in the repository, but I do want to let you know that up here we currently have experimental strategy is WGS. Now, this isn't going to only give us files with whole genome sequencing data. This is filtering only for cases that have whole genome sequencing data. These cases do have other types of data, and you'll see that that is available when we get to the repository. So our next step is to save and name the cohort. And that can be done by clicking on the save icon here, the little floppy disks, and click on save. So now we're going to name our cohort. I'm going to name it kidney male. Now, I suggest using a descriptive name for your cohort, but you don't have to tell every single attribute of your cohort just for you to know what it is. Because when I save this, there's primary site equals kidney and there's gender equals male. We have these other things, and these will all follow you throughout the portal in the query toolbar. So you will always have that information. So now our cohort kidney-male is saved, and it is our active cohort. We know this because it is in this little field here. So one of the main features that I want to bring up is cohort persistence. You can return to saved cohorts later, and they stay in your local browser cache. So if you were to clear your cache or use a different browser, that will remove the saved cohorts. And a recommendation I have now is because GDC 2.0 is in early stages. So there might be new features that come down the line that might need a reset. So we recommend saving your cohort locally. And to do this, we'll go to the next slide. Oh, the next slide here. We have importing and exporting cohorts, which can be done with these two features here. The down arrow all the way on the right exports the cohort. And this will provide a list in text format with UUIDs of each of the cases. The up button imports a new cohort. This allows you to import a file, just like the one that was exported, that can be imported too. And alternatively, if you had perhaps a list in a spreadsheet somewhere or in a text file, you could just copy and paste them in this field. And they don't have to be UUIDs like you see on the right here. They can be things like TCGA barcodes in TCGA's case, or other identifiers that aren't necessarily a unique ID. And Lastly, when you import a cohort like that, it becomes a list of cases. So you may want to keep a note of what your filters were, since it will become a list of cases and you might want to know what your query was. So outside of importing and exporting a cohort, you can also create a new cohort by clicking on the plus button to the right of the save button here. So if I click on that, you'll see that I go back to unsaved cohort. On the right here, you can see that we're back to all of the cases in the GDC, 44,451. And we only allow for one unsaved cohort at a time. So if I go to try to create a new one, this is there's already an unsaved cohort. So to get around this, all you have to do is save it, and you'll be good. Alternatively, there is the option to delete a cohort, so you can click on that, and that will remove the cohort, giving us only one for now. So the next demonstration are the GDC core tools. These are the main functions of the GDC, and these are the analysis center, the repository, and the projects tool. The cohort builder is the fourth core tool, but we just demonstrated that. So first we're going to talk about the analysis center, and to get there, I simply click on the analysis center button at the top left. So now that we're in the analysis center, you'll notice a set of cards that are labeled core tools and analysis tools. The core tools all have the descriptions there, but to see the descriptions for the analysis tools, just click on their titles. You'll see underneath the names, there is a number of cases. So we have 123 cases in total in this cohort. The clinical data analysis tool, all 123 cases can be used. However, gene expression clustering can only use 120 of these cases. There must be 3 cases that don't have gene expression data in our cohort. In order to use any of these tools, all you have to do is click on the play button next to each, or potentially the demo button if available, if that's what you're looking to do. Next, we're going to talk about the repository, which can be reached at the top bar on the far right, or by pressing the repository button in the analysis center. And something you might be noticing is, yes, this cohort bar at the top and the query bar right below that will follow us throughout the portal. The cohort bar can be pinned and unpinned using this button, and this is the option to keep it on the screen pinned as you scroll down, or keeping it on the top, and when you scroll down, you won't see it anymore. So we're going to keep this pinned for now. And now that we're in the repository, this is the place where all of the files that are harmonized with the GDC are available for download. There is a repository in V1, so it's a comparable tool. However, there's one main difference I want to show, and this applies to all of the different tools in GDC 2.0. On the right here, you'll see that there's a total of 7,834 files, which is much fewer files than you're probably used to when you go to the GDC V1 repository. And this is because these are the files that are associated with these 123 cases in your cohort. If you would like to change this and you would like to see the files associated with more cases or additional cases in a different cohort, we recommend going back to the cohort builder or changing your cohort. So our goal is to download the whole genome sequencing BAM files for our active cohort. And we can do that by going to the filters on the left here. And excuse me 1 second. Apologies, the office had some unexpected visitors. But so we like to filter down for the whole genome sequencing BAM files for our active cohort. So we'll go to experimental strategy, whole genome sequencing, and data format equals BAM. Now here we have our set of 332 files, and we would like to download these. So we can download them by clicking on the Add All Files to Cart button. And now we can see next to the cart icon, there are 332 files. We can click on Cart, and now we have a breakdown of the files by authorization level, which all of them are currently unauthorized because of their BAM files, and that requires dbGaP access. Then they are broken down by project, which is listed here. At the bottom is a list of all of the files in the cart, but I'd like to show you a couple more important functional buttons here. So now that the files are added to the cart, we can download those files using the Download Cart button. The Download Cart button has the option Manifest or Cart. Now, Cart will download the files in a tarball to your local machine. So I recommend doing this only if you have less than 5 gigabytes of data. Manifest Manifest will download a manifest of all the files, and those can be used in the GDC client or GDC Data Transfer tool. Those are both the same thing. We're not going to show you how to use the client in this particular webinar, but there are other webinars and lots of tutorials to do that, so I'd recommend looking at one of those. The button next to the download button is the download associated data. If you download a bunch of files from the GDC, you will need to know which cases they're associated with, which clinical data are associated with those, and all of that other metadata that comes with files. So there are many different options here. We have clinical and biospecimen data, TSV and JSON, in those two formats, whatever you're comfortable with. I do want to mention that because TSV is such a flat file, they are they will be provided one file per data type. So you'll get a zip file full of different TSVs. JSON is more nested, so more things can be provided in one file. The sample sheet is a TSV that has the very commonly requested fields associated with each file. So things like the ID, the sample type, the sample ID, those kind of things. I recommend checking that out if you're looking for a starting point. The metadata option downloads all of the metadata that we have for each file. This comes in JSON format. It's very nested, but it has all of the data. The remove from cart buttons also allow you to remove all of the files or just the unauthorized files from your cart. So the next tool we're going to go to is the Projects tool, which can be reached from the Analysis Center or by going to the Projects button at the top toolbar of the portal. So go ahead and click on that. And the Projects tool is one of the only tools in the GDC 2.0 that does not apply your active cohort to it. It will show you all of the projects no matter what until they're filtered down. So our goal here is to create a new cohort with only projects that include cases with primary site kidney and are from the TCGA program. So looking at the table here, you can see that lots of projects are associated with multiple disease types and multiple primary sites. But let's filter that primary site down. So we'll go ahead and type kidney, select that. We now have 16 projects, which is much fewer than last time. And now we need to filter down but to the TCGA program. So we'll go down, click on TCGA, and now we have our 4 projects here. The disease type and the primary site are kidney for 3 of these or the primary site is kidney for 3 of these projects. But one of them has 13 primary sites. This does include kidney, but it includes a lot of other things too. So these projects won't be filtered down to just the kidney cases. You will have all of the cases for all of the projects. So I'm going to go ahead and click on, I'll select these projects, or you can click on a button at the top, and this will select them to save a new cohort. So we're going to go ahead and click Save New Cohort, and this will prompt us for a name. So we're going to say 4 projects, and we're going to click Save. So now it gives us the option to set this as our current cohort, which we can do, or we can select between the two. And we're going to stay on the kidney male project because that's what the demo will show. So next, we're going to demo some of the new GDC analysis tools. These are the analysis tools that are new with GDC 2.0. This includes protein paint, gene expression clustering, oncomatrix, and sequence reads. So first we're going to go to protein paint, and we're going to get there by going to the Analysis Center, scrolling to the analysis tools, and clicking on the play button next to protein paint. And as you'll see, there are 8 cases in our cohort that cannot be used with protein paint. So go ahead and click the play button, and the first thing it does is prompt us for a gene. And our goal for protein paint is going to visualize missense mutations on the VHL gene. So we're going to go ahead and type VHL here, hit enter, and it will bring up the VHL gene. Now, along the bottom, this is our gene, our polypeptide chain, and each of these lollipops here represents a somatic mutation. The number on the lollipop represents the number of cases that have the same mutation, and if they are stacked, that means that there are multiple different mutations in the same locus. So if you were to click on 2 here, you'll see that we have 2 different samples that are that have this particular mutation. And in the selection screen, you can select the disco plot. This creates a circular plot of the genome and the locations of the somatic mutations in the genome. So right now, it's only showing mutations for the cancer gene census genes, which is only 9 out of the 77. We'll go ahead and uncheck that, and that will show all 77 of the mutations. Might be a little crowded, but if that's what you need, you have the option. If you'd like to download the image of one of these, you can click on Download Image. Going out of here, if you remember, our original goal was to visualize missense mutations on the VHL gene, and the colors here represent the consequences, and the blue one is the missense one. So as you can see, it says missense right here. But you can also see that on the legend below. So if you only want to see missense ones, you can hide these ones, or more efficiently, you can click on missense and click Show Only, which will show only the missense mutations on the VHL gene in your cohort. This this graphic can be exported as an SVG or as a reference gene sequence, whatever you would like. Also, if you were to select some of these cases, you can create a cohort from them. You'll see many different options in GDC 2.0, and I am not even going to scratch the surface. There are many ways to create a cohort in GDC 2.0. So keep that in mind. And if you think there should be a way to create a cohort, there probably is. And if there's not, let the support team know. So next, we're going to talk about gene expression clustering. So to get there, we're going to go to the Analysis Center, and we'll click on Gene Expression Clustering, which is the fourth from the left. Also note that these cards won't always be in the same place. If you were to change your screen size or anything, they will shift around as needed. So we're going to click on Play, and it loads them up. But in order to tell you a little bit about what the gene expression clustering looks like, I have this graph right here. So our goal is to visualize expression data for the VHL-associated genes, along with age at diagnosis and primary diagnosis, which are two clinical attributes. So here we have columns that represent cases. You can see that they are labeled with case IDs, and the rows represent genes that are labeled with the gene names. The lower gene expression values are blue, whereas the red gene or the higher ones are red, and white is in the middle, closer to 0. Each of these cells provides a link to the case and the gene entity pages. So if you were to click on one of these, you can click here to see the case page, and on the gene to see the gene entity page. Just like that. So going back to the Analysis Center, we have our analysis clustering, and our goal here, if that I said at the beginning, is to visualize for these genes along with these clinical attributes. So let's first talk about what this case set means. And of course, as you probably guessed, these are the cases in your cohort. So that's how the case set is created. You can zoom in based on the dendrogram, or you can zoom in with this function right here with this nice slider. And there is a good hand button here I do recommend that you can use to, slide around and look at some of these zoomed in values, since sometimes looking at it from a distance isn't so easy. But going back to the pointer, let's talk about the gene set. So these are the top variably expressed genes, but we can change them to be whatever we would like them to be. Well, whichever genes have a gene expression value. So we'll go ahead and click 100 genes, or it's going to say the number of genes, whatever you have. And we will change the gene set by clicking on Edit Group. And here we have a big old list of genes. These are the ones that are currently in the dendrogram. And we have a few options. One is to load the top variably expressed genes. This is what we have already. So you can always go back to that. Alternatively, let's clear this out. We can add genes ourselves. So we can type in something like TP53. It'll allow you to choose whichever one you want. Let's choose the regular one. There's also the option to load pre-created gene sets from msigdb, which is an external source. So here we want the VHL associated genes. So we'll go ahead and type VHL into this field here, and we'll click on this first option, which gives us a pre-created list of genes. So we'll go ahead and click Submit here. And there is just a short list of genes, because there's much fewer than before, with our cases at the top. Now we also want to add age diagnosis and primary diagnosis to visualize along with this. So we can add clinical data by going to the top, clicking on variables, and since what we wanted was age at diagnosis and primary diagnosis, we will go to diagnosis-based attributes, click on this term, age at diagnosis, and scroll down a little bit. Primary diagnosis, which is here. And now you'll see it says submit two terms. We'll go ahead and click that. And as you can see on the bottom, we have the age of diagnosis and primary diagnosis plotted here. And there's a legend for each underneath that shows you that this particular one is an adenocarcinoma, clear cell adenocarcinoma that's blue. Orange is papillary adenocarcinoma. And the age of diagnosis you can see is the same across the board because it's we already filtered by age of diagnosis, so this would be expected. So next we're going to talk about the sequence reads um, tool, which you would get to by going to the Analysis Center and choosing the sequence reads tool, which is the third from the left on the bottom row here. So if I click on this, you can see I am not logged in. I click on this and it will not let me do anything, which is okay. I shouldn't be able to do anything because you require dbGaP access to view any of these because it is viewing GDC aligned BEMS. So what we have here is a an open access demonstration that will show some of the features here. And what we're looking at is we have all the options to visualize the data. Um, we have gen code markers to show where each gene is located on the genome. The read properties, if you click on each read, it will give you properties about that read, including its pair, different quality scores, etc. The alignment coverage is plotted here, so you could show where each alignment or which areas are covered more than others, and the quality scores are present for all of the different read alignments. So next we're going to talk about the returning GDC analysis tools. These are the analysis tools that were part of GDC 1.0, but are returning in GDC 2.0 within the cohort-centric workflow. So a lot of these different tools, while you may have used them in GDC 1.0, they may work a little differently. So I recommend you stay tuned and check out some of these demonstrations. So the first one we want to talk about, so we have mutation frequency, clinical data analysis, cohort comparison, set operations, and BAM slicing download. So first mutation frequency, we can get to it from the Analysis Center by clicking on mutation frequency on the right here. We'll click on the play button, and our goal here is to find the most commonly mutated gene in the cohort, plot the survival of the mutated versus not mutated. And by that, I mean the cases with a mutation in whatever this gene is, and the cases without a mutation in whatever this gene is. And if you look at the two graphs here, that kind of gives us exactly what we need. The most mutated gene is VHL, which we have 38 out of 115 of the cases affected here. There are 8 cases in this cohort that are not part of this analysis. And then we have a survival plot. The blue curve represents the VHL not mutated cases, so cases without a mutation in that gene, and the orange curve represents those with a mutation in the gene. The gene below this is a list of genes, but this can turn into a list of mutations by clicking on the mutations button here. You can toggle between the two, and these are the 592 somatic mutations that are in this particular cohort. This is a low number because these are filtered by only the is cancer gene census filter. If you turn that off, you'll see there are many more. Yeah, 7.5 thousand. And you can filter by things like impact or type of gene on the left panel here. So that will change this table and the associated graphs. Also, if you have custom genes or mutations you're interested in, you can add them here. And one thing I want to mention is that this is another opportunity to create cohorts based on mutated genes or specific mutations. So if you wanted to create a cohort of just VHL mutated genes, you can click here. And this will add VHL to your cohort, and then you could save that or save as or whatever you need. But we're going to remove this for now. For anyone who used V1 before, this is the data that was on the exploration section of the data portal. Next, let's talk about clinical data analysis. So let's go ahead to the Analysis Center. Oh, we can save this. If we go to the Analysis Center, we can go down to Clinical Data Analysis, click on the play button. And right here, we have a lot of different graphs that are there by default. These toggle buttons can be used to remove or add them. And our goal here is to visualize occurrences of adenocarcinoma versus the other cancers. So I'll remove some of these. And here we have primary diagnosis, and we have 4 of them here. So one of the main features of the histogram here is that we can customize the bins. So we have 2 different types of adenocarcinoma, but I'm just curious about adenocarcinomas in general. So I can customize bins, edit them. So then I can take these 2, group them into a selected value, which I could name whatever I want, adenocarcinomas. And then I could save bins. And now I just have adenocarcinomas. And then these other 2 cancer types. Yeah, this can be done using different ranges, which I'm not going to demonstrate here, but here's how they can be modified with categories. So the survival plot can also be customized to compare categories within your cohort. So if you wanted to look at survival, you just click on this icon next to the histogram, and you can compare the adenocarcinomas with the renal cell carcinomas. Let's go back to the next tool, which is cohort comparison. And the goal here is to visualize survival plots between 2 cohorts that we created. So we'll get to the cohort comparison tool by clicking on cohort comparison at the analysis tool section here. 3rd from the left, we'll click the play button. And now we need 2 cohorts, and we actually do have 2. We have kidney male and we have 4 projects. So our active is kidney male. We will select 4 projects to compare it to, and we'll click run. Now it's pretty self-explanatory what's going on here. We have kidney male is the blue one, and 4 projects is the orange one. And you can see the survival plot is a bit more fleshed out with the orange one since there's many more cases. But those are these are the 2 survival plots, and then different comparing all of the different attributes of these of the 2 cohorts are below. So for example, you know that the blue active cohort has been filtered for males, so it would have no females in this particular instance. And this could be done for all of these different clinical attributes. But the next thing I want to get to is the set operations. Set operations find cases that are in both created cohorts. So we can get to this by going to the Analysis Center and clicking on Set Operations. That's an option, but we can also go from here in the cohort comparison and open the Venn diagram. So now we have in the active cohort, kidney male, we have 48 unique cases and 75 that are shared with the 4 projects cohort S2. So you can click on these, and then they can be saved with this plus button as a new cohort, which we're not going to do here, but that is an option, or they can be downloaded as a TSV. Here we have the option to save them as cohorts because we're starting with cohorts, but there is also the option to use gene and mutation sets, which can be generated from the mutation frequency app. Lastly, I want to talk about BEM slicing download, which we can get to by going to the Analysis Center, going down to BEM slicing download, which is the first one on the left here, and clicking the play button. But as you can see, we don't have access because we are not logged in. So I'm going to show you a demo, and it's actually a pretty straightforward process here. The first step is to select a BAM file in your cohort. So you'll logged in, and you can enter a search string. You can give it a file name, ID, or a case, and you can also browse 1,000 available BAM files. So it'll give you a list, and you can click on one. So that's the first step. The next step, once you get that all set, you know which file you're using. You'll want to tell it which coordinate to slice from the BAM. This allows you to download a specific region within a GDC aligned BAM file, and you need to specify which region. So your options are the area around a specific variant, which is in that case a gene or position. So this will be a gene name or a set of coordinates. Or lastly, there's an option to download the unmapped reads. The unmapped reads are kept by unmapped reads are kept during alignment so that the raw files can be retrieved from a GDC aligned file, but these are available to isolate and download on their own. So that is the end of the demo section. I'm going to go back to the PowerPoint and show you a few more things. So we're going to talk about now tutorials, guides, and support for GDC 2.0. So there are user's guides for all of the GDC 2.0 tools, and you can get to them by going to docs.gdc.cancer.gov. Click on Data Portal, and once you click on that, you'll see a list of all of the different tools that I just talked about in this presentation. Tutorial videos, including the ones that I couldn't show directly, like BAM slicing or sequence reads, are available as video guides here. So you will be able to watch a video on how to use any tool that was mentioned here. Those will link to YouTube, and you can watch them there. They're approximately 10 minutes, a little more, a little less. So yeah, I'd like to plug those tutorial videos. And lastly, if you have any questions about GDC 2.0 or anything about the GDC really, or any feedback about GDC 2.0, we would really love to hear it. And here is our support email, support@nci-gdc.datacommons.io. You can send it to that. This is also all available on by clicking on the send feedback button or by going to gdc.cancer.gov. So thank you all for your attention, and are there any questions?
I
Oh,
think there is one question, Bill, in the chat about the recording. And what we typically do is once the webinar has been complete, we do process the recording for compliance, and then it does get posted on our GDC website. So Bill, do you want to show where it gets posted on our GDC website?
yeah, for sure. So here I am. I need to get some WebEx things out of the way. So you can get to that by going to gdc.cancer.gov. And On the right here, you can see the support menu. You can click what monthly support webinar, and you'll see all of the different videos are here. You'll see whatever the upcoming webinar is. This is the one we're doing right now. And the past webinars, the videos available here, and you can link to YouTube directly from this. So you can do that. I see that there is a mailing list that we have. If you go to support, you can join the mailing list um, under, sorry, it's under contact us. Oh, here, sorry. Join the GDC user listserv. So I'd highly recommend you sign up for that with any news. Um, we'll send any news about the GDC through that. And before the um, the help desk can be reached by going to by sending an email to support at nci-gdc.datacommons.io. So that is here. Just by going to gdc.cancer.gov and clicking on support.
Okay, Bill, it looks like there's two questions. The first is there a tool to visualize protein expression data similar to the gene expression clustering tool?
So we don't have a tool that does that yet. Something you might see if you go to the repository is the protein expression data, which is reverse phase protein array. So we do have the data, we just don't have any tools to visualize it yet.
Okay, and the next question is, are there any changes to the GDC BioLinks R package?
Um, so you'll have to ask so it's the TCGA BioLinks group are not affiliated with the GDC directly. They use our API and they'll sometimes contact us with questions, but I just want to mention we are not affiliated with them, so I'd recommend reaching out to them with any questions you have and for any support with TCGA BioLinks.
Okay, and there's a question, what would be the pipeline to analyze the RPP expression data in the absence of a tool? Would it require coding with the API?
So you can download all of the data directly through the portal. You won't have to use the API at all. And they're pretty small files, so I'm pretty sure you'd be able to download them directly through the portal. Um, you I'm not sure however you would like to analyze them. I'm not sure what that would require, but all of the metadata that you would need, you could also download from the cart page, like I showed before. So if you were to click on cart, all of the metadata and such would be available here. So you wouldn't have to design any API calls or anything like that. You would just go down to download this stuff, and you would have it on your machine. I see another question. I'd like to ask if it's possible to change settings for building a clustergram for gene expression analysis. As an example, the distance among samples or the linkage. Um, so I don't think the gene analysis tool does that yet. We can definitely pass that on to the group that develops the gene expression tool. Um, but for now, it only visualizes the most commonly expressed genes versus the cases and plots a matrix accordingly. Okay, so here we've been using Python to query the REST API of the GDC. Do we need to make any changes moving forward? So, not to my knowledge. I think the API, if you've been using the features that existed previously, those should work the same. There are some new things that will be added to the API that will be outlined via documentation. But to my knowledge, anything that worked for V1 in the API should work with V2. If you come across anything that doesn't, please let us know, but it should all work the same. Okay, are there any more questions? If not, I'd first like to
There's one more question.
oh,
They wanted to uh, download in clinical data only. Only the partial barcode is given, so it is sufficient to download data, or do we need the full barcode?
yeah, that's a good question. So if you have clinical data would be associated with a case. So if you're using TCGA data, um, the barcode, let's pull up the cohort builder and look at those cases. Um, so we can go to a table view. This is a feature I didn't demonstrate, but this is something you can look at all the cases that are in your cohort. So if you had something like this, TCGA-E9-A1NF, you'd be able to download whatever clinical data was associated with that because anything downstream would refer to a sample or a portion, analyte, aliquot, those type of things. And clinical data is associated specifically with this entity right here. Now, if you wanted a specific RNA-seq run or something, you might need to find the aliquot barcode. And you can get to that. Well, I don't know what you're looking at specifically, but if you were to go to a case ID, sorry, this computer is chugging a little bit. If you were to go to a case here, let's go back to the table view. Look at that. Okay, so I'm not sure what you have in your end, but if you were looking for something like RNA-seq, you would see something like this would be a full aliquot barcode, and that might be what you need. Sometimes you can get away with just knowing the case because sometimes there's only like one aliquot that's tumor or whatever. Oh, but I see a follow-up here. Um, oh no, there's not. Sorry. But anyways,
Bill, I was going to suggest if you have only the partial barcode, you can put it in the search, and usually it will try to find the rest of it.
yeah, that's a good idea, Alonzo. I like that. So like, let's say I'm removing one of the last character from that. So let's see what that gives us. I remember it was a 6. So it gives you other ones, so it might not be the right one because they are all unique. So this one would be the correct one. But so yeah, if you only have partial barcode, you might need a little more information. But I guess the
It
answer is
gets you closer and does work with the aliquots and samples too, not just cases. Yeah.
yeah, that's a good point. So if you had a partial aliquot barcode, that might get you where you need to be.
Okay, there's the question. Is there a way to quickly filter by project within the repository tab?
So within the tool itself, no, but I can show you kind of a shortcut to do it. It's not exactly what we have in V1. And also I do appreciate the feedback um regarding that. So anyways, let's close this up a little bit. And let's go to the repository. So now we're in here. Um, Within your cohort, so if you had a cohort that had everything, you could open this up. Um, you don't want table view. This would be summary view. And you can filter by project here by choosing the selection view. And I can search for just CPTAC 3. And as you can see under the repository, this will filter in real time. So you can see, let me see, here, scroll over here. These are all CP Tech 3 now. So as a workaround, if you were to go to like create a new cohort, let's go to our unsaved cohort, which should have everything. If you open up this tab here, you could filter down by, let's say you wanted Apollo or something, or your example is TCGA UVM. So if you go to project TCGA dash UVM, click on that, and we have TCGA UVM here. So that is a workaround. Yeah, that feature isn't exactly built into the repository, but there still are ways from the cohort toolbar to do that. And I think I was kind of rambling a little bit, so let me start from the beginning. This left button here, I didn't demonstrate this too. This discards any unsaved changes. So you can click on that, and I just made some unsaved changes. So now we are back to all of our cases. So you go to access additional cases, project, selection view, search values, TCGA, UVM, select that, and now our repository is only TCGA UVM.
Okay, there's a question on how to request access to BAM files.
Yeah, so that requires an ERA Commons ID and dbGaP access through that ERA Commons ID. So we do have some good videos on how to do that. You get there at gdc.cancer.gov under support, multi-support webinar, and it might have been under frequently asked questions. I know some of these have obtaining access to controlled data in the GDC. January 31st, 2022. It still should be up to date, so I'd recommend checking this out.
Yeah, and Bill, you might want to point them to the web page on obtaining access, which has a video from dbGaP.
Yes, that is a really great video. Highly recommend that. Here it is right here. So if you go to access data, obtaining access to controlled data, which is where we are, scroll down a bit, and you'll see this video. This is from dbGaP. This wasn't produced by GDC, but they do such a good job showing you how to do it that we recommend anyone who's looking for dbGaP access looks at this. Thank you, Sharon, for posting that in the chat. Okay, so the last thing I would like to show you, we still have a minute or so. If you were to click on send feedback here, we have that's a good way to get to the help desk and video guides. If you have any questions about any of this, all of the tools I just showed you. Here's the one on Cohort Builder. Here's the Analysis Center. All the different analysis tools are lined up down here in this table. And these all have YouTube links, downloading with the repository, viewing projects. All of the different tools are here. So feel free to watch them. We recommend checking them out. And yeah, if there aren't any more questions, thank you all so much for your attention. We appreciate it, and we hope you all have a good time using the new GDC 2.0. Thank you.
Yeah, thanks, Bill, and thanks, everybody, and we really value your feedback. Thanks so much.